Okay, hello everyone. My name is Frithjof Gressman. I'm a PhD student advised by Lawrence Rauschberger. And online joining, we have Ivan Rykov. He will introduce himself in a second, and he's actually going to start off this talk. So we're going to present our work on efficient multi-objective optimization of neural dynamical systems through joint learning of objectives, constraints, and sensitivities. Very long title, and hopefully we'll explain this over the course of the talk, what this actually means. So Ivan, feel free to get going.
Hi, everyone. My name is Ivan Rykov. I'm a research engineer at the Schultes Lab at Stanford University. Frithjof and I have been working together on a optimization package that can be applied to some of the problems of the Mine in Vitro project. So our broad aim is to utilize our knowledge of neural circuits and build some type of computational system, broadly speaking. And so we imagine that given some experimental systems, such as the MEAs developed by Mine in Vitro, from which we can record electrical and optical signals, such as field potentials, calcium imaging signals, and so on, we can build algorithms and systems that can translate neural signals into some signals that are useful for computational systems. And in addition to that, we have created the Mine in Vitro simulator, which we use to create computational equivalents of the physical neural cultures. And we also use them to try to understand the nature of neural computation. As in the simulation, we have access to individual neurons and the components of the system that we wouldn't have experimental access to. And then in terms of thinking about computational systems, we know that neurons can perform some forms of pattern recognition. Networks of neurons can process sequences and can recognize and generate sequences. And we hope that perhaps with primitives such as pattern recognition and sequence processing, we might be able to build logic such as state machines and similar, which are the building blocks of computation. And so with that lofty goal in mind, we have started developing a number of biophysical neural network models that aim to recapitulate the functionality of the Mine in Vitro neural cultures. So we build models that are based on biophysical neurons that consist of distinct morphological compartments such as dendrite, soma, synapses that process the input signals, then the dendrites integrate them, and then the spikes are generated in the axon. And of course, we have a number of detailed models of molecular mechanisms that express the synaptic and ion channel dynamics, and then that allows us to build biophysical network models. And so there are a few advantages in this, that we can have a good operational understanding of neural computation. We can generate testable predictions that are useful for experiments. These models somewhat reflect biological constraints and do not require a lot of training data because they're based on physical principles. But there are a number of disadvantages as well. One of the main disadvantages is that there are significant uncertainties in the parameters that are obtained through experiments. In neuroscience, as many of you know, experiments can be done at the single neuron level or at the slice or whole brain imaging, etc. But usually, if we need neuronal parameters versus network parameters, we have to obtain those from different experiments. So the parameters to constrain our models come from different experiments, so which adds a lot of uncertainty. And the parameter landscapes themselves are rather complex. The biophysical models are highly nonlinear dynamical systems. And so even small changes in parameters can result in large changes in behavior and overall dynamics. And so thinking about our sort of target application of building a logic with neural system requires even when we have biophysical neuron models and maybe models of circuits that can perform some simple pattern recognition functions. So in order to scale up to finite state machines or more complicated logic, that would require the interaction of a lot of neural circuits. And we need to, even with our basic understanding of neurons, we still need to learn and understand how the circuits will interact and how to build hierarchical complex logic systems. And so we believe that the framework we have been developing for surrogate optimization would play a big part in solving these problems, in allowing us to understand complex multi-scale neural circuits. And so just to give you an idea of the parameter searches that will be involved. So first, exhaustive parameter searches would be basically intractable for many neuronal models. Here on the right is a model of the CA1 region of the hippocampus that was previously built in the Schultes lab with hundreds of thousands of biophysical neurons, multiple synapses, and so on. So even if we consider every connection to be one parameter, still that makes exhaustive searches are out of the question. And here in the table below, I have included, just to give you an idea of the scale involved. So for a single neuron model, maybe we have on the order of 10 to 20 parameters, relatively quick evaluations. But as we increase the network size and the number of parameters, the computation required increases exponentially. So in order to evaluate even a moderately sized network, we now require tens of minutes on multiple cores and supercomputers. So efficient optimization and efficient directed parameter searches are very important when dealing with these systems. Okay, and just to give you an idea of the overall surrogate evolutionary optimization algorithm, it consists of an initial sampling phase and evaluation of the model system. Once a sufficient number of samples is obtained, the algorithm builds a surrogate model, and I'll explain in a moment how this is done. And then the surrogate, which is much more cheaper to evaluate computationally, is used in an optimization algorithm. We've used a family of evolutionary optimizers. Once the evolutionary optimizer finds the best parameters of the surrogate model, then we go back to the original model and we sample it with the parameters we have found, which then gives us an improved surrogate and so on. And so we need essentially a search strategy that minimizes expensive simulation, but it builds by building an effective surrogate model. Importantly, and here I think Frithjof will show several important results, but this optimization strategy goes just beyond just more efficiently finding the best values. Once we have a sufficient number of samples of the system that we're trying to optimize, we can compute gradients and perform sensitivity analysis, which actually can reveal key properties about the neural network or the model that we are studying. And so I think Frithjof will show that this infrastructure goes beyond optimization and allows an iterative process of hypothesis generation and experimental testing. And so we have the software package we call DMOSOP. It consists of a number of libraries for several sampling algorithms, several published optimization algorithms that we have also modified and adapted for surrogate optimization, a number of analytical tools for analyzing the solutions that have been found, and then a number of surrogate modeling methods, primarily methods that involve Gaussian process regression, various forms, and a model developed by Frithjof that he will talk about next.
Yeah, so basically we've seen that we have this optimization framework very generally that implements all sorts of state-of-the-art methods. And we now implement a specific model that we call joint surrogate modeling. And I'll basically go through and explain what that means and why it's helpful for our specific problem. So when we want to optimize our MIV system, basically what we're modeling mainly is a system like this. It's a two-compartmental model of the motor neurons that we're using in the experiments. So basically we have a soma compartment, a dendrite compartment, and then all these different ion channels. And as Ivan mentioned, the problem is often that we have a lot of these low-level parameters, like conductances of each of these channels, all these electrical properties that we fundamentally don't have data for. Like we don't know how each ion channel is really constituted in the real life. What we do have access to is usually higher level observations. So for example, we might have experimental data where you inject a current, increasing current, and you observe how does the firing change. And you would expect that as you increase the current into the system, the firing goes up. And so the challenge is really try and find these parameters such that we basically recapitulate the data that we see in the real system. And the actual challenge that we've sort of addressed in this work is that we're not only interested in solving the objectives, so basically matching these two things, we also need to do that in a way that honors all the constraints on the system. So for example, it would be very implausible if all the points are matched, but it's in a zigzag line. So there's sort of constraints on like the monotonicity of these functions. There's constraints on like the plausible physical ranges of these parameters. And so that actually is making this optimization much more interesting and problematic, because it's not only just a matter of sort of minimizing the objectives, but also honoring these constraints effectively. And to show how much of a problem this is, so this is basically doing a random search, basically naive sampling, 50,000 samples on this motor neuron model. And we are plotting basically for each of the constraints, how much the percentage of the simulations we're doing actually ends up meeting these constraints. And as you can see, it's very low. So this is in percent. So basically less than 1 in 1,000 simulations in the beginning actually ends up being sort of valid. So we're basically throwing away, we're running 1,000 simulations and only one is really giving us anything of value. And so what ends up happening is that basically often you can't even optimize these systems if you don't first find a method to effectively sample in a region that actually meets all the constraints. And so this is really the problem that we're trying to address in this work. Basically, the question is, can we model these sort of systems in a way where we integrate the objectives and constraints at the same time in one surrogate model? And usually, people are using Gaussian process models, but they have two problems. One is that they're very hard to scale. So as the size increases, computational complexity goes up quite significantly. And then more fundamentally, it's very hard to actually make a joint model in regression and classification context. So it's very hard to make the objective and constraints work at the same time. I'm not going to go into the technical reasons for that, but yeah, it's just a fundamental thing about Gaussian processes. And so the idea of this work is to say, maybe we can actually use conventional neural networks that don't have that limitation, because for conventional neural networks, it's very easy to scale them up. And importantly, it's almost trivial to basically integrate constraint, classification, and regression at the same time. Why don't people use neural networks in this context? Well, the reason is that it's extremely difficult to make this work with little data. Neural networks are known to require a lot of data. But in this work, we basically show that if you're clever about it, you can make this work and reap the benefits of using these net systems. So I'm going to give a brief overview of the architecture for those who sort of have knowledge of this, but it's not essential to understand. But the key idea to make this network, so this surrogate model work, is to say, instead of doing the regression directly on the parameters, we basically do a pre-stage of transforming the inputs with a multi-head attention mechanism. And so basically we're taking a pre-processing step before we do our regression step. And then to make this work, we apply a special output normalization, which allows us to basically ensure that the model doesn't get overconfident, even in a regime where it has very little data. And again, I'm not going to go into the technical details of it. If you're interested in it, go read our paper or talk to me. But a lot of work went into basically making this work for small amounts of data. And the key thing here is that now we have this ability to basically model objectives and constraints in a single model. And I'll show how this is useful. OK, so let's look at the actual results. As I said, we want to basically get to a point where we can find realistic dynamics. And I'm just going to plot some here. And the first problem is actually even figuring out how do we even know that the solution is good. The reason why this is a difficult problem is because what usually happens in these systems is that we have multiple objectives that we want to meet. So for example, I mentioned the current injection and frequency. So that might be one objective on this axis. But we might have other objectives at the same time. So we want to maybe keep the decay time in a plausible range. And now what often happens is you optimize and you find two solutions, one being very good on this objective, another one being very good on that objective. And so it's actually not trivial to sort of generally say which model is a good solution to this problem. There's a vast literature out there on how to do this, and we're just basically using this. So one of the metrics we're using is the inverted generational distance. And the idea there is basically that if you have a solution that you know is good for this problem, then you can basically compute Euclidean distance to your found solution to know if it's a good idea. The problem with this, of course, is that you first need to know the solution to the problem. And often we don't have that ability. So what we do in this work is we basically let our optimizer access a lot of time and compute to obtain like a very good solution. And then we use that as a reference point to evaluate our cheaper approximations. And I'll show in a second how this looks like. But so this is the first metric we're going to use in the results is IGD. Basically, think of it as the lower the better. The closer you are to the target, the better. And then the second metric we're going to use is called hypervolume. And the neat thing about the hypervolume is that you don't need to know the actual true metric. How does that work? So basically what you do is you take the worst possible solution that you can think of. So basically a solution where you know that any solution you will find will be better. And then you basically draw a line of all your solutions you found in blue. And now you compute the volume. I mean, in this case, the area, because it's two-dimensional, but you compute the volume of sort of the area that you capture here between the worst possible solution and the best possible solution. And the idea is that if I now find a new solution, this green one here, I can basically see that the volume increases as I basically get closer to the unknown optimal one. So for the hypervolume, the idea is basically this. I don't know what the true one is, but I know if something is better or not. So for the hypervolume, which is the second metric we're using, basically think of it as higher is better. And the absolute value doesn't mean anything, but we can compare between solutions. OK, and so with these two metrics, we're now going to go and start sort of exploring these models. And we applied this very broadly, and not just the mind in vitro single cell model, but actually for a bunch of different cell types in the hippocampal system. So all these are different cell types, different names. It doesn't really matter but the first thing to do is basically establish a baseline, which is sort of just randomly sampling. So we are basically evaluating different sampling strategies here to see in red how well are we actually solving this problem by just basically trying randomly. And you can see there's a wide variety. So for example, for NGFC, you get basically nowhere by trying randomly. Like you're literally not making any progress on the hypervolume. And then other problems, particularly the ones at the top, it's actually, you can get pretty far by just trying them randomly. So, and then we have the Gaussian process regression, which is sort of the gold standard in the community. And you can see there's also varieties, basically. Some benchmarks are completely solved by that. Other benchmarks, not so much. OK, so this is our baseline. And now we're going to go and apply our model. And this is the result that you would get. So what are we showing here? So basically at the bottom and at the top is the amount of simulations we're running. So basically how much compute are we spending by evaluating these systems. And then on the y-axis is the normalized hypervolume. So higher is better. And now we have the baseline, which is basically not doing any surrogate modeling, which is this gray line. So that's the default. And then we have all these different surrogate baselines. And you can see that we are now able to basically much quicker converge to a similar solution. Okay, so if we look at this in basically in terms of what is the solution that we find, and we compare this to our optimal solution with IGD, we can see that our models are sort of very close to the optimal, outperforming these more conventional baselines. And so this is just for one cell. And importantly, what we really are interested here is saving the compute. So what you can see is that if I can basically stop after 5 epochs, and I'm going to get the same result more or less than if I had run without this optimizer. So basically across all the models, you see that you can save up to 80% of the required simulations. And that's a nice result because even if your simulations are getting larger, this will always hold. So if my simulation is twice the size, I'm going to save twice as much because we're basically just shaving off simulations that we don't have to run. And we can now go and do this across the board. And we see that basically across all these cell types, we get similar results, except for the bistratified cell. And I'm going to talk about this. But in the bistratified cell, you see this is one case where actually you would be better off running without surrogate. So something here is going wrong. Basically, the surrogates are not able to find the correct solution. And now we can ask questions about the system. So basically, one of the hypotheses we had when we started this work was to say that like it's helpful for us to model constraints and objective at the same time. And so now in this plot here, we're trying to understand if that's actually the case. So what we're modeling here, we're basically comparing across all problems by ranking each model, so which model comes first. And then we rank them, so which one is globally the best model, right? And you can see that the Gaussian root process models that are commonly used are at the bottom. But then interestingly, our feature transformer is actually better than like a basic ResNet neural network. So this confirms that our feature transformation approach makes sense. And then more importantly, you can see that if we give the model access to constraints and objectives, which is denoted as C+O, it actually performs better than if you just give it access to the objectives. So that basically confirms our hypothesis that modeling these things jointly is a good idea. And then when you do the same to understand basically how well are we going to find solutions, you get a somewhat surprising result that basically if you have enough compute and you don't care about efficiency, you might as well go and just optimize the objective and ignore the constraints. So you actually you're better off just spending the compute just optimizing objectives if you had like enormous compute. So basically what this means is that the constraints are not useful to find good solutions. They're basically useful to tell us where the good solutions are. And that's intuitively makes sense. You really, instead of like being stuck in a region where you don't want to be, the constraints basically tell you very quickly where you have to go. But if you had the compute and then you might as well not bother and just use a single model. Yes. Yeah. So for people online, the question is, what if you mix them, basically use the constraints first? And yeah, that's right. So we actually have results in that too. So you can do that. That's right. So you can basically exploit this and do what you say, basically use the constraint in the beginning and then switch to your optimization. Yes, it does help. Like it works. Yeah. Yeah, it's a good question. Okay, and as Ivan mentioned in the beginning, we're not just trying to just save compute, we also want to understand things, right? And so one of the nice things about these surrogate models is that, like, if you have the true problem, which is basically this super complex simulation, it's very hard to understand what's happening. I mean, and now if you have the surrogate model, it's kind of more manageable because it's sort of a simplified version of this thing. And so basically what we can now do is we can, because we have this differentiable neural network, we can ask questions about what our system actually does. And I'm going to show one result here. It's a very busy graph, but basically what we are showing here is on the x-axis all the different parameters. And then we are looking at the relative importance of these parameters for minimizing the objective and minimizing the constraints. So basically, a way to read this is basically to the left are the parameters that are sort of the most important that you really care about when you want to minimize your objectives. And what's nice about this is that basically when we do this analysis, we find that, for example, this calcium parameter is apparently the most important parameter. And that actually tracks with our biological understanding of these systems because the calcium channels are one of these channels that have a huge influence in the short time scale of changing things. And, you know, Ivan can tell you more about the neuroscience behind it, but basically it shows that we actually, just by applying this method of, you know, trying to optimize it, we can now go back and ask questions about the biology. And it seems like that the surrogate model actually learns something about the biology and not just the basic optimization problem. Yeah, so it's closely related. So it's basically the gradient with respect to each parameter times the input. So it's, yeah, you can think of it as sensitivity. Matter of fact, we actually have a sensitivity result. So for you, so that's actually, so for everyone who doesn't know, sensitivity is basically a way to tell which parameter is important and which parameter is the most sensitive. And there's a lot of methods in the literature to analyze systems in general to find out which parameters are important. The two methods that we implemented are FAST and DGSM, and very common methods in the literature to use this. And what we now can do is we can basically compute our surrogate gradient to get a similar similar estimate of the sensitivity. And what we show here, so at the bottom, again, the IGD metric, lower is better. So we can show that basically by computing the gradient on our surrogate model, we actually match the performance of these sensitivity methods. So basically, before, if you wanted to do sensitivity, you basically have to run the simulation again and do a different method. Now we get this all in one model. Same for the hypervolume. So this is a nice way of saying that we basically have one model to rule them all objectives, constraints, and we can also use them to analyze the sensitivity. Yes, yeah, so that's a good point. So basically the question is, what's the difference between the importance for the model, like for the biology, and then the importance for the optimization? And actually, the point is that basically both things are true. So if a parameter is important for the biology, it's very likely also going to be the one where you have to be very careful when you're optimizing it, and vice versa. So they are basically the same. So basically, that's what this result is saying, is that if we compute the sensitivity with respect to the optimization objective, we get a very good proxy for which ones are actually importantly sort of in the biology of things. Yeah, it's a good question. So the question is basically, does this hold in general, or can there be cases where the optimizer deems a parameter very important, but it actually isn't that important biologically? And I'm pretty sure there are cases like this, and maybe Ivan can comment on this. And that's definitely a limitation. So we're not claiming that, if our method says this parameter is important, it's necessarily going to be biologically important. What we are showing here is basically just a correlation and saying, actually, we are able to pick up on these underlying biological systems. But yeah, it's a good point. So we could study that further in the future. Okay, and then the neat thing is that now we can actually use this sensitivity to basically help our optimizer. So we can basically go round trip. Now we say, okay, if we know which parameters are important, we can actually feed that back into our optimizer and tell the optimizer, look, this parameter is very important. Spend your samples here. Don't worry about the other ones. And so if we do that, so this is again the bi-stratified cell that I showed earlier, where basically previously the methods failed to match the non-surrogate baseline. But then actually, if we incorporate the sensitivity information that we have available in the joint model, then as you can see, we again find that the surrogate model is now outperforming the baseline. And the reason why this is important is because you might say, well, why go through all the trouble? I mean, let's just run it on a supercomputer, right? It's cheap. So, but the thing is, so let's say we have 3,500 simulations to run. We can obviously do that on a single cell, and that's why we are, in the paper talk a lot about single cell optimization, because we can show all these great trade-offs. But reality is that once you move to larger scale systems, you're probably going to only be able to run 1,000 simulations maybe on a supercomputer. And so then it really matters that you are able to do these sophisticated tricks, because if you only have 1,000 simulations to run, it doesn't really matter that this thing is ultimately better. You really want to be getting there quickly, and that's where the constraints are helpful. And so with that idea, I like to close and just show that basically talk a lot about Simia cells, but as I said, we also have the ability to do this now on a network scale, and we use data from Xiaochuang and Xiong, and that they published in their paper to basically recapitulate some of the firing distributions. And so now basically there the objective becomes minimizing the KL divergence between the firing distribution that we see in the experiments and the firing distributions that we see in the simulation. And that's where basically we can't run 10,000 simulations, and where this method sort of is now helping us to overcome this. We can't really prove it because we can't run the large scale model at this size, but there's reasons to believe that this allows us to achieve network optimizations that we wouldn't be able to achieve without this approach. Okay, and with this, I'm handing back to Ivan to close, to basically give an overview of where we think this will be heading now that we have this established.
Okay. Okay. So actually, I wanted to go back. I'm sorry about it. Okay. So Frithjof went through a lot of biophysical details and how we really try to understand the dynamics of individual neurons and simple circuits. But we would like to eventually do more sophisticated computations, going back to the idea of performing pattern recognition with biophysical neurons and scaling it up to perhaps performing some logical operation. And so the infrastructure that we've built, the Mine in Vitro simulator and the optimization infrastructure, allows us to now start building these first simple pattern recognition experiments where we have some signal that is encoded with spikes that is presented as input to a biophysical circuit. And that also allows us to incorporate different neural features, such as more sophisticated dendritic models, synaptic models, and so on, and to really start understanding how each of those features contribute to neural information processing. And hopefully this will give us some idea of how to build logic systems. And so just to give you an example, here is on top is a input signal which is just two-dimensional, consists of two sinusoidal inputs. which then are discretized and represented as firing rates based on receptive field models. So we assign 4 input neurons to represent each dimension of the input signal. And the color, the intensity here represents the firing rate. These firing rates are then produced stochastic input spike trains. And then the bottom is the response of one of the neurons that Frithjof has optimized, provided the input. So this infrastructure, of course, can scale to networks. Here is a slightly more complicated example with a 3-dimensional signal. In the middle are the spike trains generated that represent the input signal, and then the response of the network. And here the network is just consists of excitatory neurons, so we haven't added any complexity to the circuit. But with this framework and with the optimization, now we can run optimizations where we have set the objectives to be certain information processing metrics, such as accuracy or mutual information or entropy and so on. So we hope that we can translate the optimization infrastructure to really allow us to build functional circuits. And I will show, so this is a very sort of artificially constructed example with pattern recognition with a small network. The inputs are, again, sine waves, 100-dimensional, so slightly larger dimensionality with a few hundred samples. And I have evaluated, these are very short control runs, and I have not yet applied Frithjof's joint model, but we hope to do so in the near future. But I have evaluated a simple architecture of excitatory neurons that are connected to each other, as well as a circuit with inhibition, with feedback inhibition. So there are inhibitory neurons that receive input from the excitatory neurons. And using 2 types of encoders, the receptive field and phase-based temporal encoder, the accuracy, so we can see that the training accuracy for all these models is similar, but the test accuracy tends to be a little bit better for circuits that involve that have feedback inhibition. These experiments are too small of a scale to really draw any big conclusions, but I think this is the beginning of a sort of a framework that would allow us to explore multiple architectures, both in terms of neurons and synapses, as well as network connectivity. And I really hope that with the models developed by Frithjof, we could also analyze and compute surrogate gradients and really try to understand why one architecture performs in a certain way for certain tasks. And so with that comes our conclusion. So what started as a sort of black box optimization of neural systems now has led to the discovery that surrogate models are not only effectively reducing the computational load for optimizing large networks in complex models. We are actually now able to understand how the different parameter regions are explored, and we think we have a powerful method for exploring neural computational architectures. And I think that's now it's time for more questions.
Thank you. Oh, what the reference was? Yeah, so the reference point is basically if the idea is that so we showed basically the plot where we basically run without the surrogate model. But because when you're using a surrogate model, you're evaluating less, you actually cut off very early. So what the reference point is basically if you do surrogate, like non-surrogate optimization, but you let it run the full length of it. So it's in the order of like 100,000 evaluation versus in this case, we only look at the first 1,500. So we're really giving the optimizer a lot of compute time to find the best solution. And then we compare against that solution. So it's a bit like we're comparing against a 1,000 times compute budget. Yeah. No, exactly. So that it's just for us to measure and convince ourselves this is working. In a real setting, particularly for networks, we don't know what the best solution is. So we will never know, basically. But this is just to demonstrate this, yeah. But the hypervolume, as I said, that's the good thing about the hypervolume is you don't need to know the solution in advance to tell how good your model is, at least in relative terms. Yeah, so, okay, for the people online, basic question is, can you not estimate how well, like where the optimal hypervolume is? And the reality is actually you can't. The reason is that you would imagine that like for a problem where you know that the best solution is where all objectives are zero, that might be the case because then the hypervolume is just taking up the whole space. But there are a lot of problems, particularly in this area, where basically you can find a solution where basically the more progress you make on one objective, the less you make on the other. And you can't fundamentally not trade them off. And in that case, you just don't know what the perfect hypervolume would be. But this is a good question. So you can estimate, but it's not, fundamentally, it doesn't work. Yes, that's right. You can only compare relatively. You don't know if you're doing great, basically. Yeah, sure. Thing. Oh, yeah. So there's actually multiple ones. So for example, the one I showed is basically matching the FI curve. So that would be one objective, basically, how well are we matching the current frequency relationship? And that's one objective. And another one is the difference in how close is the time constant to a reasonable value and that sort of thing. Yes, for example, yeah. But it's up to you how many ones you want to put in. I think in this case, we put 4 different objectives. So, but yeah, that's a bit of a question, like how do you optimally design them to get good results? And that's basically mainly, that's why basically Ivan comes in and just tells me this is how we're going to do it. And I'm like, fine. So yeah, there's a lot of questions around that. But so yeah, so it's again a question of what to do. What I did in this case was I fit the distribution, so get the PDF of the firing distribution of your data, and then I'm computing the KL divergence with the simulated distribution in like yes, over sort of for each MEA channel globally, exactly. Yeah. So, but you can do simpler things. You could like just add, like go for the mean and standard deviation across channels, something like that. It's up to you, but yeah. So that's, to be honest, that's actually something that isn't settled yet. What's the best idea there? Yeah. 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 Yeah, this is a good question. So basically the question is, all these optimizers already do some form of sensitivity optimization. Why bother? And I think it could well be that like your NSGA2 would be able to pick this up, but we just empirically don't necessarily see that. It seems to be the case that you're better off explicitly integrating that sensitivity information. But it is a bit of an unclear question why that is. I think my hypothesis has to do with the way the neural networks smooth the loss surface. Because one of the things you can do, you can actually do direct gradient descent on your surrogate without ever running NSGA2, for example. But I find that that works less well. And I don't really have a good explanation for why. But it's a good question. So yeah, it could well be that it's not necessary. And so all we are claiming here is basically that we're matching these sensitivity methods. If it's a good idea in general, I actually don't know. Cool. Any other questions? Okay, well, I guess then it's time for pizza. Thanks, everyone.